Swayam Prabha. Digital India. Educated India.
Okay, so as I said, today we are going to address in some detail a very central concept in synthetic biology. There is the concept of chassis, or chassis, as French say. So what this word or this term evokes is something that comes from car engineering or manufacturing, and it's this idea that one can have a basic, say, frame with a number of functionalities. In the case of cells, metabolic functionalities, genetic functionalities, regulatory functionalities, on top of which we can add parts and devices in and out, maintaining the main frame, but just altering the modules that we put in and out. Okay, so this is a very strong metaphor, a very strong image of what we are after in synthetic biology. So just to put a frame in the discussion we are going to have today, remember this motto. So motto, I don't know if you remember, is this kind of short sentence that uses to be in national shields and this type of, say, banners and these things, like saying, well, this is something that we are convinced that is, this is the shield of Spain. I say plus ultra, that means much farther. And well, you can forget about that. Every country has a different motto, or every company has a different motto. In our case, we have three major mottos. Please remember these mottos for the rest of your life. Parts make devices, devices make systems. Parts, devices, systems. Part, devices, systems. It is something completely, central to synthetic biology. Then, and this is something that we have also discussed in previous days, the program, the software, is different from the machine that reads the program. So the program in biological systems is encoded in DNA, and the machine that reads the program is different from the program. That's quite important. And also, and keep that for the rest of the talk today, the chassis is different from the implants. There's a principle of modularity. So one thing is the chassis, and a different thing are the implants. Okay, remember that. Okay, so this would be the ideal, say, scenario that we have for synthetic biology constructs. So you have a chassis model where you have all the basic functions, genetic functions, biochemical functions, physiological functions, and then you have models that you engineer and that can go back and forth. Now, in the best of the worlds, these models would be completely independent, completely orthogonal, as we discussed in previous days, but in reality, there's always an interplay, an interaction between the models and the chassis, and between the models themselves, in such a way that the behavior of every model is influenced by something called retroactivity from the chassis, and then vice versa, the models can also influence the behavior of the chassis. Okay? So you have models, we have interactions. In the best case, we would have orthogonal models, but you will see that this is not always possible. And then you have this phenomenon, or this idea of retroactivity that we are going to discuss a bit. Why do we have these interactions? Well, number one, because if you remember a few days ago, we discussed that the models and the host of the models compete for cell resources. And therefore, there is this competition that makes that the behavior of the chassis and the behavior of the models is not completely independent. But also, and this is quite important, cells are intracellularly not homogeneous. So that means that even if we want to have a model implanted in the genome, then the products of the model will go into the three-dimensional space of the cell and will interact with other, say, components. And therefore, there are many effects that are connected to the fact that cells are not internally homogeneous. Instead, they are heterogeneous. There's a considerable degree of what is called intracellular granularity. Okay? So all these circumstances make that there's a phenomenon called retroactivity between the chassis and the implant, in such a way that you have an input that goes into this particular device and it has an output, then this output will get somewhere, and that somewhere will lash back into the functioning of this model, and you have this, say, non-perfect functioning of the whole design that you have there. So you can make an abstraction and decide, well, imagine that, for instance, you have like two devices here. You have an upstream component with a transcriptional factor, a promoter, and a product X, for instance, that will influence or will activate expression of another promoter. So in the best of the worlds, then depending on the configuration of the various modules, you can have a very homogeneous input-output, say scheme. But sometimes when you have this retroactivity between the model and the context, then instead of having something perfect, then you have something that is quite imperfect. Okay? So in this model, for instance, you have just a self-regulation, and under given parameters, this self-regulation will give you an oscillation. But if this component is influenced by the context, then you have all these perturbations in the otherwise very even, say, a scheme of input and output that you have here. Okay? So in practical terms, well, not in practical terms, this sometimes is difficult to really predict how this is going to happen, but you can abstract what happens in terms of having a main model here that in this case could be the chassis itself, and then you have all these other modules that you can give parameters in for the different degrees of retroactivity. Okay, and this is something that if you like that, then you can make a model and then see to what extent implantation of a model will influence the performance of the other or vice versa. Okay, so this is something that just one has to keep in mind. Well, the cellular context. I mean, you have metabolites, you have proteins, you have supercholine, you have what you may call physiological circumstances. Okay, and this is what alters the behavior of the model, and you know, the term context is very vague, I know that, but in general this is used to describe all types of circumstances or material circumstances where the model that we engineer performs. Okay, so let's continue with this, and then you can think in terms about the chassis. Well, you know, the plural of chassis can be chassis or chassis. In reality, it doesn't matter. Okay, so this is what we saw before. We have at the level of the program, because at the end everything is under control or is encoded by DNA. Then you have the genomic chassis. You can have a genetic implant, and then what you are dealing with here is with a DNA with composition of the chassis and the composition of the genetic implant. But just keep in mind that this scheme has a projection in the other side in terms of having consequences or repercussions in the machine. So a genetic implant will encode a new trait. The genomic chassis will deliver or will hold the entire biochemical and structural, say, structure of the cells. And then at the end, what you have is something that you can detect that is a functional or metabolic phenotype. Okay? So for the most part, we basically can manipulate this part, the program, with expectation, or I mean, we can manipulate or we can study this part with expectation that later we will see something happening at this side. Okay, so these two sides are different, but keep in mind that the program and the machine that reads the program are different, they are separated, but keep in mind also that the machine can go without a program, but the program cannot go without a machine. Okay, so the DNA by itself will do nothing. It always requires a machine to read it. That is the entire machinery of the gene expression flow. Another important circumstance is in terms of the context that you mentioned before, is this idea that genes and regulators are not in the air, they are surrounded by a very well-connected proteome, and the proteome itself is always influenced by the metabolism, okay? So this is exactly what you were asking about, the context. So one thing are the genes, and we would like to play only with genes, but then it happens that the physical molecules of DNA, where the genes are encoded, are in the context of a proteome, and everything is in the context of a metabolism. So you have to imagine the cells, you know, like a space where you have many molecules acting at the same time, not only genes but many other, say, actors. Okay, so why this idea of chassis is so important? Well, let me just explain you the following. At some point, we have a wealth of DNA all over the place coming from metagenomic studies, from the environmental microbiome. I mean, the amount of DNA that you can get in different parts, in different niches, in different locations is immense. So, and this DNA is held both by a so-called culturable fraction, so the part of the microbiome that can be taken to the laboratory and grown in a petri dish or in a bioreactor. But just remember that there's also another big, say, share of the entire environmental microbiome, the entire, say, life on Earth, that is not culturable. Not culturable means that the DNA is there, the cells are alive, but you cannot take them to the laboratory and grow them there. This is a classical problem in contemporary microbiology. There are many bacteria that are there, are very active, but we don't know how to take them to the laboratory and have them growing. And both this fraction and this other fraction has interest in DNA, okay? So if we want to exploit the functionality of the DNA that is encoded in the entire, say, planet for different applications, then we cannot rely in many environmental bacteria as they are there. So some of this DNA is born by some bacteria that cannot be taken to the laboratory. So then the idea is that we take the DNA and we put the DNA into a recipient that eventually should be able to express what you have in the DNA and deliver some type of interesting activity. So this DNA can be put in different hosts and then mine activity, improve it by evolution, improve it in some cases by orthogonalization, and we discussed yesterday. And at the end, you may have a good functional model. And there's this functional model that can also come from, not only from bacteria, but also from animals or plants or being entirely synthetic, may eventually, or we may want eventually, to put it to work in a chassis. This chassis has to originate for our own sake, for our, say, convenience. It has to be something that is culturable, that is something that can be brought to the laboratory and made grow in the laboratory, because if not, it will be very complicated. So at the end, what we have is a situation where we have members of this community here. In many cases, the best are those that grow fast, and then by making some manipulations, and I will explain to you some of them later, then you produce a chassis that then would be the perfect, say, starting point for having a good catalyst for different applications. So the way to have chassis can imply like two say, domains. One of them is the domain that is connected to what you may call familiar biochemistry. And if you work in the, say, realm of familiar biochemistry, you can start with a natural bacterium, for instance, and then in this bacterium you can have plasmid, transposons, viral vectors, no problem. You can enter some modifications into the genome of this bacterium, and then you have an streamlined or edited genome. I will show you later an example of that. Or in some cases, you can even have a bacterium that we manipulate to lose completely its genome. So at the end, this can be, there are methods to get rid of the chromosome, and then at the end we have a kind of vesicle, a kind of genome-less cell, where we maintain all the biochemistry that we had at the beginning, but without no DNA. Okay, so these are the different choices that we have as synthetic biologists to really run a given experiment or a given project. Also, we can go a little further and then have one of these bacteria with orthogonal ribosomes. That means ribosomes that are able to decode non-natural or alternative genetic codes, and we mentioned that in previous classes, or even express proteins with non-natural amino acids. But even at this stage, we are in what could be called familiar biology, okay, where you have DNA going into RNA and going into products. But synthetic biology enables to go one more step, and then to move into the side of completely artificial cells that are biological, but the biology is not familiar. And it's not familiar because the nucleic acids are not the typical DNA, are xenonucleic acids, where some of the bases or of the bases are variants or alternatives to the bases that appear naturally. The structure of the DNA is also different, and at the end, despite these differences, the molecule still contains biological information. And well, you can think also on different types of information-bearing molecules or genomes that have a different chemistry. Okay, so in this way, one can speculate, and there's a lot of research going on in that respect, that we can produce in the near future, perhaps life forms or something that you identify as life forms that is based in a different type of chemistry. And this is an endeavor that is being run by many laboratories because it's a kind of, it will be a super big thing that we are able to create life in the laboratory with a different chemistry. Okay, this is not science fiction. This is something that belongs to the agenda of contemporary synthetic biology, not for an application, not for a biotechnological application, but for kind of understanding what is life and how, what are the principles that make that enable a transition between a system that is not live versus one that is live. Okay, but in our more, say, realistic domain, that is the domain of biotechnology, then our objective, remember, the main agenda of synthetic biology is about programming biological entities. So we start with something that is wild, then eventually we want it to be completely engineered, but in between we have another step that is very important, that is domestication. Okay, so what is the difference between a wild animal and a domesticated animal? Can anyone tell me what you think are the major differences? I mean, there are more than one difference, but can you propose like what? Yes.
They don't attack or harm humans.
Exactly. So that would be one aspect, that they are not dangerous to us. More.
Selectively bred for certain traits like intelligence towards humans, or in the case of dogs, you can say even appearance.
Okay, that's a good point. Any other aspect?
They obey us.
Obvious.
They obey whatever we say. They will do. We can make them do whatever we want.
Exactly, that's the point. So the difference between a wild animal is that the wild animal will not obey your orders. The domesticated animal, in principle, will obey your orders, even though we know that sometimes it does not obey our orders. Okay, and the idea is that we have the idea of programming is that you have a biological entity that will follow our orders, similarly to what we have at, I don't know, like a kitchen device, and you just put the washer and then it works, right? So that would be the idea. So we are not there completely yet, so we will see in this talk how far we are in the attempts to domesticate biological systems. As a matter of fact, the history of domestication is very interesting, and I don't know if you ever thought that there are many types of animals, but in reality there are not that many domesticated animals. Okay, so only about 20 species have been domesticated throughout history. Okay, so the cows, the turkey, the sheep, whatever. But there are some animals that, despite numerous historical attempts to domesticate, they have never been domesticated. Monkeys, for instance, you have monkeys all over the place here, you don't have them as pets, right? Because they don't follow our orders. I mean, they give us a very hard time in many cases. They have their agenda and forget about trying to domesticate them. We have foxes, for instance. Foxes are beautiful animals. However, despite numerous attempts to domesticate them, no way. For instance, zebra horses. You know, zebra horses are these animals in Africa that have like all these strips in the body. They are super robust and so on. And you know, intuitively, you would think that they would make perfect replacements for horses. Yet, after millennia, they could not be domesticated. So that means that domestication is a genetic trait. Some species have the kind of tendency or the predisposition to be domesticated, where others do not have them. Now, you can make the same argument for bacteria or for microorganisms. So some microbes are easily domesticated, like E. coli. You know, E. coli is the best domesticated, or one of the best domesticated microbes, or yeasts, and you know, yeasts are all over the place, or lactobacteria. But there are others that might look interesting, but they cannot be domesticated. And there are even some pathogens that have very interesting genes that would benefit from. However, they have never been domesticated, and it's very difficult that we can domesticate them. So the idea is that maybe we should start with thinking on having a limited number of species that can be domesticated for the sake of synthetic biology. But there's a part of this idea of having this chassis in connection to domestication. There are other considerations. So we need so why do we need to define what is a chassis? Okay. So I'm going to go through various texts, some legal texts that have been produced recently, and well, there are various, say, reasons why there's an urgency and a necessity to define what is a chassis as part of our interest in having good semantic standards. Okay. So, one of them is that the word chassis now is applied to many bacteria. And in my opinion, and the opinion of many, this is wrong. I mean, you cannot call a chassis any bacterium. You have a bacterium of any type, you make a mutation here and there, and it's a chassis. No, it's not a chassis, and therefore we have to qualify what is that. The other thing is that there's, for a long time, before synthetic biology, there have been around a large number of hosts of recombinant DNA, okay? Yet, being a host of recombinant DNA does not make you to be a chassis. You just remain a host of recombinant DNA. And therefore, there's this necessity to have a criteria to differentiate genetically modified organisms from what we propose to design as synthetic biology agents or chassis. I will return later. So, but one of the most important motivations for really trying to define what is a synthetic biology chassis is that in legal terms, the legislation and the regulations in biotechnology for using genetically engineered biological items are very well established, and the idea is that synthetic biology is different from traditional genetic engineering, and therefore synthetic biology produce chassis and produce synthetic biology agents. And how do we differentiate these synthetic biology agents that are produced through synthetic biology versus what you may call traditional products coming from genetic engineering? So that's why we need a good definition of chassis. So a definition of chassis, and this is a kind of definition, is an engineerable and reusable biological platform with a genome encoding a number of basic functions for stable self-maintenance, growth, and optimal operation, but with the task and signal processing components growingly edited for strengthening performance under pre-specified environmental conditions. Well, it's a kind of long definition, and every word that you find here has some kind of meaning. A chassis has to be a biological platform, in many cases macrobes, but they can be of other types, that have to be amenable to engineering, to genetic engineering, can be reused, and they might contain a genome with basic functions that enable growth and optimal operation under specified environmental conditions. Well, this definition is not just the definition of Victor de Lorenzo, it's an official definition by the EFSA. EFSA is one of the international regulatory agencies for food safety in Europe, but they have equivalent agencies in other countries and in other, say, territories. And what I just share with you in this definition is like the shared opinion of what is a chassis. So, well, it's just to reproduce verbatim the definition of chassis in the EFSA documents, okay? But there are some disclaimers that you have to make in this definition of chassis. So the term chassis can be applied to many types of platforms, not only bacteria, but also plants, different types of animals in principle, and even cell-free systems, namely systems that are not say, alive proper. However, the definition that I just mentioned applies only to live microorganisms. Also, sometimes computer people use the term chassis just for some in silico models. Okay, but again, this is not say the definition that we have just discussed. Now, the key of the definition is performance, no minimized genome. So that you have a smaller genome does not help at all to define what is a chassis. Because what we are focusing is on performance. Okay? So a given chassis is optimal for a given performance, regardless of the size of the genome. Although in some cases, you can remove unnecessary functions, and you will see later. And also, and this is important, there will be not just one chassis, but there will be successively improved versions of the same chassis depending on how science develops. So by the same token that you have iPhone 1, 2, 3, 10.5, whatever, and in computer program you have the same, you have different versions, then for the chassis should be the same. Namely, that you have a variant that you give a, like Pseudomonas putida 2.0. But then will be 2.1, 2.3, or Pseudomonas putida 3. I mean, you can have that code to really indicate successively improved versions of the same chassis. Now, I'm going to spend a couple of minutes, well, a few minutes, going through some definitions and some nomenclature, okay? So you have to be aware of something that is important, and is that the jargon, the nomenclature, the standards that we use in general are difficult to implement, are difficult to be accepted by the community. And there's this famous sentence by this gentleman called Murray Gell-Mann that was a Nobel Prize in Physics that say a scientist would rather use someone else's toothbrush than another scientist's nomenclature, okay? Meaning that sometimes we propose a semantic standard, and that semantic standard may or may not be accepted by the larger community, okay? Okay, so just to understand what we are talking about, let me spend, I say, a few minutes to tell you a few things about what words we use in synthetic biology and what do they mean in a rigorous fashion, okay? Okay, isolate. Well, remember that we are mostly talking about bacteria and microorganisms. Well, an isolate is a naturally occurring microorganism, and well, that they have properties of fundamental or applied interest. An isolate, no problem. Variant. Okay, so a variant is a naturally occurring microorganism with defined changes in its native genome, because it carries changes in the genome that have happened either spontaneously or entered through RNA technology, through recombinant DNA technology. So we have an isolate and we have a variant, okay? So we have, I don't know, a penicillium strain that produces a penicillin, and then we have a variant that hyperproduces penicillin. That's still a variant. We are there. Now, what is recombinant DNA? So recombinant DNA are DNA sequences from biological systems, either amplified from existing genetic material or synthetic, that are propagated in the same vectors. And this is the material for construction of devices and implants of all types, okay? So remember, isolate, variant, and recombinant. So, but there are more definitions here. Now, what is a recombinant DNA host? A recombinant DNA host is a non-pathogenic variant, and we use variant using the definition before, capable of exogenous DNA capture and manipulable with genetic tools. So for being a recombinant DNA host, you need to be non-pathogenic, you have to be able to capture or maintain exogenous DNA, and you have to be manipulable with genetic tools. If you don't fulfill these conditions, you are something else. You are not a recombinant DNA host. And, well, what type of recombinant DNA hosts are there? Well, you have a plethora of them, the most famous E. coli, but you have others, okay? Now, what is a genetically modified organism, GMO? It's a recombinant DNA host that we define here, plus minus modified DNA in cis or in trans. What do we mean by cis or in trans? Cis is when we execute the changes in the chromosome itself, in the genome, and trans is when we place the implants in a separate piece of DNA, for instance, in a plasmid, okay? So for instance, one E. coli strain carrying a plasmid will be a GMO, because then it will have the strain that is modified with the specifications with given recombinant DNA elements. Now, what is a chassis? And remember the definition we did before. A chassis would be a recombinant DNA host that is optimized for deploying genetic devices under specified conditions, okay? So what is the difference between one and the other, of this recombinant DNA host and a chassis? That the recombinant DNA host just takes a DNA with no, say, modification of itself, whereas the chassis is optimized for deploying the activity that is encoded in the genetic devices. And well, as I mentioned, and visually I showed with the car and the chassis and so, is that a chassis is the biological frame where recombinant DNA components can be plugged in and out for creating new functionalities. Now, what is an implant? So an implant is any genetic synthetic DNA sequence added to the chassis genome and encoded one or more biological parts. And these implants would typically involve a number of parts with a given relational logic for forming devices and circuits. And be patient, we are just getting to the end of this definition. What is a synthetic biology construct? A synthetic biology construct will be a synthetic biology chassis plus a genetic implant, either in cis or in trans. So in many cases, in the recombinant DNA technology, you can put your devices either inserted in the chromosome as one more chunk of DNA in the chromosome, in the genome, or they can be added to the cell in trans by means of a plasmid or by means of an extra chromosomal element that in most cases are plasmids. And as in bioconstruct, most frequent result biooperations. Now, what is a standardized chassis? Because we talk about chassis, but what is a standardized chassis? I will return to that. It's a barcoded chassis that fulfills other criteria. I will return to that later. But then at the very end, we have a synthetic biology agent. A synthetic biology agent, in properly talking, is a standardized synthetic biology chassis with genetic implants, either in cis or in trans. And out of all this definition that I have told you, this is the final product of synthetic biology, okay? The one that goes not only to laboratories, not only to play with them for projects, for research projects, but the one that goes to industrial and environmental applications. So to the ultimate, say, deliverable of synthetic biology is to have consolidated biological materials for biotechnological applications, whether industrial or environmental, okay? So that's an important consideration. Now, how do you become a chassis? Okay, and again, this is a quite dense slide, but I think that it contains very interesting information. So, well, to start with, there's not a absolute defined break between GMOs and synthetic biology agents. So at some point, you start with 100% GMO, you end up with 100% synthetic biology agents, and the decision of where you put the limit is not crystal clear. However, you can indeed define what is involved in having simply an RNA recombinant host to have a simple SynBio chassis and to have a standardized SynBio chassis. And again, I insist that the good stuff is have a standardized SynBio chassis. And you will understand later why it's important to have a standardized SynBio chassis or a number of standardized SynBio chassis. Well, for RNA, having an RNA host, a recombinant DNA host, all you need is to be able to uptake exogenous DNA, that you have some genetic tools, and that you are not virulent. And then you are happily an recombinant DNA host here. But then how do you move from here to here and you become a synthetic biology chassis? Well, you need then to know much more about the platform that you are talking about. You need to know about genetic stability. For instance, you may decide whether you want to have homologous recombination or not, and to what extent having that will have an impact on the durability of the genetic implants that you use. You need to know what type of energy metabolism you have. You need to know what stress resistance you have. You need to know, I mean, of course, you need the sequence genome. You need to have very clearly determine what is the percentage of external DNA that you have there. Also, you have to know very well what is the sensitivity to antibiotics and to phages. And you have to enter into the genome some degree of editing for efficacy in targeted scenarios. We will spend some time, one of the classes later, dealing with genome edition. But just for now, take for granted that we have the tools to edit the genome and to enter modifications that increase the efficacy of these bacteria in the targeted scenario. But then we get into another territory. And this territory is that of a standardized SynBio chassis. That is in reality what regulatory agencies and the industry wants, okay? And then in that case, you may want to have a defined antigenicity. You may want to have a degree of containment. We'll return to that later. You want to be able to trace where you are, what is your identity in the middle of other bacteria. You need to know to what extent you can give DNA or take DNA as a donor or as a recipient from the environment. You have to know what is the impact of implants in your general metabolism, in your general physiology, and then you may come up with this orthogonality index. And eventually, what you want to have for applications, real applications, is to have a safety qualification. This safety qualification, there are two types. In the American qualification, you have this kind of a stamp that is called GRAS, that means generally regarded as safe. And then in the European system, you have this QPS that means a qualified presumption of safety, okay? So once you fulfill all these things, then you can use your chassis happily in a large variety of environmental and industrial conditions. Okay, you may say, well, nice, but who are the chassis? Give me examples of chassis. Okay, so in this slide, I show you a few, well, about 1,000 chassis that are interesting for different applications. Remember, I told you that by the same token that you have many animals, but we have domesticated only about 20 animals, 20 animal species. We have millions and millions of different types of microorganisms, but it might be a good idea not to try to domesticate all of them, but to have a limited number of them domesticated or converted into a chassis. Let me just mention some of them that are very important in the world of synthetic biology as well-accredited chassis and why they are so. First of all, you have mycoplasma. So mycoplasma is a small bacterium that has a small genome, and well, it's not terribly interesting from the point of view of biotechnology because it's difficult to manipulate and so, but it has the advantage that to this day is the life system that is better studied and where every single molecule has been mapped, and we know a lot about this mycoplasma, where a whole synthetic genome has been entered for the first time, and also it has some kind of side interest, and is that it's an inhabitant of our lungs, and in some cases it has been proposed to be a vehicle for delivering therapeutic activities to our lung. Okay? Now we have E. coli. No need to explain why E. coli is so interesting. It's a laboratory workhorse. It's a good recombinant DNA host, and you have a whole bunch of genetic tools. Then you have Pseudomonas putida. You will hear about Pseudomonas putida later today. And the advantage of this bacterium is that it's tolerant to environmental insults, and it's a good platform for metabolic engineering. So see that in this list we are covering like different applications, different aspects of contemporary biotechnology. Bacillus is a nice bacterium. It's also a laboratory workhorse, and it's easy to recombine for various genetic manipulations, and also is a very good secretor of proteins and other things into the environment. Corynebacteria is a Corynebacterium is a species or is a type of bacteria that are very good for large production of amino acids in industrial settings. Saccharomyces cerevisiae, I don't need to explain you that yeasts are very important in biotechnology. Pichia pastoris is a type of yeast that has the ability to secrete to the medium large amount of recombinant protein. So this is also a very good chassis. We have this super interesting new member of the whole list, that is Methylorubrum extorquens, that is a very good chassis for one-carbon pathways, and one-carbon pathways are involved in capturing CO2 and convert it into biomass. So this is, from an environmental point of view, can end up being a very important chassis. Then we have some cyanobacteria. Cyanobacteria are interesting because they can grow out of CO2 and light, so they don't need more things. And obviously they are very, very interesting for metabolic engineering and for CO2 fixation. There's a tremendous interest and massive research in cyanobacteria in the last few years because of these properties. Streptomyces is a famous platform for production of antibiotics and secretion systems. Vibrio natrigens, this is becoming a super popular chassis in synthetic biology because it grows very, very fast. The duplication time of Vibrio natrigens in LB, in rich medium, is about 7 minutes. Okay, so that means, and I can really accredit that because I have it in my laboratory, that you strike a plate, you go to have a coffee, and when you return, the cells are grown on the plate. It's just amazing. Okay, so this is one of the fastest growth bacteria that you can imagine. It has some problems because it's sensitive to oxidative stress, but well, it's not too bad. You have lactic acid bacteria that is very good for engineering probiotics. Alteromonas is a nice bacterium for biodegradation of bad compounds in marine systems. Rhizobium is a great platform for targeting plant roots and for nitrogen fixation. Yarrovia has become a very popular platform in the last few years. It's also a yeast, and as its name indicates, lipolytica, that like fat, that can grow in an apolar, it can act on a polar substrate in an apolar medium. Halomonas has become also a very popular, chassis in the last few years because Halomonas grows in non-sterile seawater. Okay, so what is the advantage of that? The advantage is that if you have, you want to produce a product or biomass or a antibiotic or something, then you have to go to a fermenter. The fermenter has to be closed. It has to have a rich medium. It cannot be, you have to be careful with contamination and all the rest of it. However, Halomonas grows happily in seawater, and in seawater there are very few bacteria that can grow because of the high salt content. So Halomonas has become in the last few years a chassis of choice for large-scale say, production of, for instance, bioplastics, simply because you can have a big extension of this bacterium planted in seawater, and at the end you produce a lot of biomass and deliver the product that you want. So, yes?
So this yarrow via lipolytica,
Yes.
what solvent system does it grow?
Well, it grows in different, I mean, you can play with, you have water, but then you can add growingly, say, apolar components of, I mean, it cannot grow on pure toluene or in pure acetone, or in pure, but you can have a high percentage of the medium with these compounds.
It will be a mixture of aqueous and apolar substances.
Yes, and also sometimes you can have an emulsion, and then in the emulsion, this yeast will grow on the surface of the little droplets in the emulsion. Okay. So you have you can have either one-phase fermentations where you have the composition of the medium like amended or added with an apolar, say, liquid, or you may have a complete emulsion, two-phase emulsions, where you have droplets, and then the droplets on the surface will be colonized by these aerobia cells that will execute the catalysis on the very surface and the very interface between their biomass and the droplets. So it's super interesting.
Yes.
Okay. So there are various laboratories working with aerobia. It's becoming very popular. Well, you know that this is like the most famous actors and actresses in the world, right? So depending on the year, depending on the circumstances, some of them might be more popular than the others. And if you tell me, well, what are the top chassis of the last year? I would say, well, it is methylobrum, is Vibrio, and is Halomonas. Yarobia has been popular for quite a while already. Okay.
So as a follow-up question, has it been or can it be used to clean oil spills?
To clean oil?
Yeah, oil spills.
Not to the best of my knowledge. You know, I think that marine bacteria are, by the way, you're asking that because you just had a petroleum leak in front of Chennai. I don't know if you heard that.
No.
Okay, so I don't know if you read that in the press. There has been
Today.
the last few days. I don't know exactly what day, but I read that in the press. So I don't know the details, but I mean, petroleum leaks are happening all the time. It's nothing special. But when they happen, they create a big alarm because they are very spectacular. So many marine bacteria are able to do the job very, very well, and you don't need to have fungi or yeast that they have a very bulky biomass, and at the end, doesn't look good. But marine bacteria are very well able to clean up petroleum spills with much better efficiency than your aerobia. Okay. Okay. More questions on that? Okay. Good. Okay. Well. I'm sorry that you don't see what is here, but how do you, I mean, this could be a question for you. So on the basis of what we have discussed so far, what you think should be, how can you differentiate GMOs from SBs? I mean, imagine that you are legislators and that you are asked, well, we have a legislation for GMOs, and you are telling me that now you have other things that are not GMOs and are synthetic biological agents. How can I differentiate what is a GMO versus what is a synthetic biological agent? Okay, can you imagine? I mean, maybe some of you in the future will be in that situation, okay? Okay, so you have different possibilities. You can think what is the contents of recombinant DNA. Instead of bulk recombinant DNA, you can consider the percentage of parts, or the percentage of steps that are required for the construction, or the percentage of information that you have knocked in. But just remember that this is something that is under debate today, okay? And this discussion we're having today on chassis, synthetic biological agents, and GMOs is already quite alive, and maybe in the next few months or years, the things will be settled. Okay, so what is one main advantage of this approach? Well, the good advantage is that once you have determined many properties in connection to the safety and connection to the performance of given chassis, when you have a new construct, a new bacterium, a new strain, and you want to ask for a permission to implement that in an industrial or environmental setting, you don't have to start from the beginning through all the tests and all the reports and so. all you have to do is to concentrate in the implant and convince the authorities what is the impact of your implant in the pre-existing chassis that you have. If you don't have that, then every time that you have a new construct, a new strain, a new something, then you have to apply for a different type of permission. Okay, so having this standardized chassis is also a good way to accelerate the time between having something working in the laboratory and having that accepted for industrial or environmental conditions. Okay, and you have your approval, and then you are very happy there. Okay, so I think that we deserve a 5 minutes break, and then we'll continue, okay? Because there's still a lot of stories I have to tell you today. 5 minutes, okay?